In this session B of Unit 4, we will learn about entomological surveillance and how to monitor and evaluate vector control interventions. By the end of this session, you will be able to describe the key elimination programmatic questions that require entomological data, state the priority indicators for entomological surveillance in elimination and post-elimination settings, explain the main design elements of an entomological surveillance system, and explain how to monitor and evaluate the effectiveness of indoor residual spraying and insecticide-treated nets. Let's begin by discussing entomological surveillance. Entomological surveillance can be defined as the regular, systematic collection, analysis, and interpretation of entomological data for risk assessment, planning, implementation, monitoring, and evaluation of vector control interventions. Entomological surveillance is a core function of the malaria program. As with surveillance for human disease, entomological surveillance data are collected to make programmatic decisions in order to optimize vector control. In the context of malaria elimination, the following are the main objectives of entomological surveillance. First, entomological surveillance is needed to track the composition and density of malaria vector species over time and space. Knowing the seasonality of transmission permits optimal timing of interventions. The composition of vectors is needed to know the species for which insecticide resistance data should be collected. Second, entomological surveillance should be used to monitor insecticide resistance to permit optimal choice of insecticide formulations. The frequency, intensity, and mechanism of resistance should be assessed in the principal malaria vectors and, when possible, in the secondary vectors. Information on insecticide resistance is needed to choose insecticides and the type of ITN, as we learned in the previous session of this unit. Third, entomological surveillance may be important in proactive or focal investigations to understand entomological contributions to novel, continuing, or resurging transmission. For example, an outbreak of malaria may be related to changes in the vector population. Entomological surveillance activities could help determine whether changes to vector populations are driving transmission. Fourth, entomological surveillance can help identify threats to vector control. For example, the relative proportion of transmission occurring outdoors may increase as a result of effective control of indoor biting or indoor resting vectors. Vectors may be monitored to detect any significant change in the location in which transmission takes place in order to decide whether supplementary interventions are required, such as new tools for control outdoor transmission. Finally, entomological surveillance can be used to estimate levels of receptivity. In the first part of this module, reviewed the concept of vectorial capacity and receptivity on how entomological data can contribute to our understanding of how receptive to transmission an area might be when nearing malaria elimination or post-elimination. The entomological parameters considered in characterizing receptivity include the vector species, composition, and the characteristics of the vectors that influence transmission. Important traits such as biting time, preferred biting location, host preferences, dispersion, and resting behavior should be known for all the principal vectors, as these traits determine receptivity and thus guide the selection of interventions. Entomological investigations are also important in the monitoring and evaluation of vector control interventions. We will discuss this in more depth in the second half of the session. The design of entomological surveillance systems should be based around the questions that the program must address. In areas approaching elimination or post-elimination, there are several key questions. The first question is around which interventions should be implemented where in the country in order to eliminate malaria transmission. When programs are planning their malaria control interventions, they must group subnational areas with similar characteristics into strata and then determine the appropriate mix of interventions. Entomological data on vector composition in different parts of the country can help determine the appropriate vector control strategies to be used in those areas. The second question relates to insecticide resistance and asks whether local vectors are susceptible to the insecticides used in vector control, or whether there is a need to change or modify interventions because of increasing resistance. Prospective information on vector susceptibility to different classes of insecticide can assist in determining which insecticide should be used for IRS or larviciding, or whether ITNs or PBO should be deployed. The third question relates to outbreaks and focus investigations and asks what potential entomological drivers of transmission are responsible for novel, increasing, or persistent malaria transmission. And finally, once a country has eliminated malaria, entomological data can help estimate the level of receptivity and therefore the types of vector control interventions that may be needed. Another important set of questions revolves around the effectiveness of existing interventions to reduce transmission, but as these questions are related to the monitoring and evaluation of vector control activities, we will cover this topic in the second half of the session. The first question asked by most malaria elimination programs is which vector control interventions should be implemented in which areas of the country. The most important consideration for planning vector control interventions is the occurrence, species composition, and density of Anopheles mosquitoes. The presence or absence of mosquito vectors is a critical indicator. Areas without competent Anopheles mosquitoes or with very low vector densities may not require large-scale deployment of vector control interventions. Reactive rather than proactive interventions could be more cost-effective. Knowledge of the vector species composition can improve understanding of the vector's behavioral tendencies, larval habitats, and transmission efficiency, which may influence the choice of vector control. Measurement of species mosquito density over time identifies seasonality and vector abundance that can be important for knowing when to deploy certain strategies. Depending on the technique used, measurement of densities can also identify mosquito behaviors such as peak biting times and indoor versus outdoor biting. Knowledge of the key behaviors of mosquito vectors, such as whether they feed indoors or outdoors, peak biting hours, blood meal preferences, and resting behaviors can also help programs to choose the most appropriate vector control strategies and to decide whether supplementary vector control such as larviciding is needed. And finally, surveillance data that indicates whether larval habitats are present and productive can contribute to determining the feasibility of larviciding as a supplementary vector control strategy. After interventions have been chosen or implemented, malaria programs need information on whether vectors remain susceptible to the insecticides in use. For both IRS and larviciding, there are a number of different insecticide classes that can be deployed in the intervention, and information on vector susceptibility can help inform that choice. For ITNs, only one class of insecticide, pyrethroid, is currently available. However, there are new ITNs that also contain PBO, as we saw in the first half of this unit. In areas with pyrethroid resistance due to monoxygenase activity, regular ITNs can be exchanged for ITNs with PBO to renew their effectiveness. In the Surveillance Unit 5E, we will spend more time discussing focus investigations, which are implemented when there is a need to understand the occurrence of new cases in an area where no cases have occurred for some time, a resurgence or increase in cases, persistence of cases, or novel or unusual malaria species. There could be many reasons why a focus has new malaria cases, a resurgence in cases, or persistent transmission. A focus investigation should start by developing hypotheses about what may be causing transmission. Some hypotheses are more likely to be the reason for transmission than others. We have assembled a list of potential reasons for changes in transmission in a focus, ordered from the most to least likely. We will discuss all of these hypotheses in Unit 5E. However, for now, let's focus on two of these hypotheses that relate to malaria vectors. The most likely reason why transmission may be occurring in a focus is because of lapses in or inadequacy of malaria prevention or case management. This could be the result of delays in vector control or in diagnosis and treatment. But it is also possible that there is poor quality or poor coverage of these interventions. Changes in the coverage or quality of vector control is one of the most likely reasons and should be investigated early. We will talk about approaches to evaluating the quality of vector control in the second half of this module. Although less likely, there is the possibility of transmission due to changes in vector density, species composition, or behavior. If hypotheses have been refuted, or there remains a question as to whether entomological changes have had an impact on transmission, then entomological surveillance should be conducted in the focus to investigate whether there are entomological drivers of transmission. We were introduced to the concept of receptivity in the first half of this unit. Receptivity is the degree to which an ecosystem in a given area or a given time allows for the transmission of malaria. Vectorial capacity is strongly related to receptivity, and therefore receptivity can be established through entomological indicators. It would have been helpful if vectorial capacity could be monitored over time and space, but the measurement of all of these components of vectorial capacity is extremely challenging and beyond the capacity of most malaria programs. There are other methods that also estimate receptivity, such as past levels of malaria transmission and calculation of reproductive numbers. In Unit 9 on prevention of reestablishment, we will take some time to review the non-entomological approaches to measuring receptivity. Unfortunately, there is no gold standard for measuring receptivity. Because receptivity is closely related to vectorial capacity, entomological data can contribute to an understanding of the degree of receptivity. The most important principle in receptivity is that an area cannot be receptive to transmission if there are no competent malaria vectors. Therefore, entomological surveillance can contribute to classifying an area as receptive or non-receptive by demonstrating where malaria vectors are and where they are not. Once an area is known to have malaria vectors present, knowing which Anopheles species are there provides information on efficiency of the vector and the mosquito's general behavior tendencies, such as their biting preferences. For example, an area with vectors that prefer to feed on humans as opposed to animals will have a higher level of receptivity than places with vector that prefer to feed on animals. Finally, the density of malaria vectors in an area can help to categorize areas of relatively higher or lower receptivity. As vectorial capacity is proportional to the density of vectors, all things being equal, areas of greater vector density will have higher levels of receptivity than areas of low density. This table summarizes the entomological indicators that are important for answering the 4 questions of elimination programs that we just finished reviewing. The 4 key questions are shown at the top of the columns and the indicators along the left. The occurrence, meaning presence or absence, of malaria vectors is important for knowing where vector control interventions are needed. It is also a key component of receptivity because an area without mosquito vectors is not receptive to transmission. Knowing which vector species are present in an area can inform intervention choice, but species composition is also important for deciding which mosquito species should be tested to determine susceptibility to insecticides. At very low transmission, it is difficult, if not impossible, to incriminate vector species. That is, it is very difficult to find sporozoites in mosquito vectors because transmission is very low. Often the best that can be done is to infer which species is likely to be the primary malaria vector based on its occurrence in an area, knowledge of the vector's behavioral tendencies, and past incrimination as a vector. Species composition might also be important for determining the entomological drivers of transmission during outbreaks or focus investigations. Introduction of new species or relative increases in the abundance of a particular species could be a reason for increasing transmission. Species composition is also important for estimating receptivity because certain species are known to be more efficient malaria vectors than others. Measurement of mosquito density is not a priority indicator. In many very low or low transmission settings, mosquito density might actually be too low to be measured reliably. However, density might be helpful in determining which species is likely to be the primary vector. Knowledge of the primary vector can help programs to choose which intervention to deploy and which vector species should be tested for insecticide susceptibility. Measuring mosquito density periodically over a calendar year could help to identify seasonality in mosquito density, which could improve the timing of vector control interventions. In outbreak or focus investigations, measurement of mosquito density could support identification of the vector species that is most likely responsible for transmission. However, in focus investigations, it is important to remember that entomological data are being collected many weeks after the cases that may have triggered the investigation were infected. Mosquito density measurement could also help to determine the level of receptivity in an area, particularly in composition to other areas in the region. It is nice to have information on the key behavioral tendencies of mosquito vectors, but this is of lower priority for two reasons. First, there are yet no clear-cut WHO recommendations on how behavioral tendencies should affect vector control decisions. Second, it is very time-consuming and challenging to measure mosquito behaviors, particularly if mosquito densities are low. With respect to focus investigations, knowledge of mosquito behavior might be helpful to understand if there has been a behavioral shift that could be responsible for increasing transmission. Knowledge of mosquito behaviors might also contribute to a better understanding of the degree of receptivity. For example, mosquitoes that prefer human blood and tend to bite and rest indoors are going to lead to greater vectorial capacity and higher receptivity than mosquitoes without these tendencies. Habitat availability and habitat occupancy should only be measured to determine whether larviciding is a feasible and appropriate intervention, or to monitor the effectiveness of larviciding. Resistance frequency and status should be measured routinely if any insecticide-based vector control measures are implemented. If ITNs are used and resistance is detected, pyrethroid PBO ITNs should be deployed to improve the effectiveness of the intervention. Resistance assays are only needed in outbreak or focus investigations if one of the hypotheses for transmission is the possibility of vector control failure due to resistance. In some of these indicators are high priority, whereas others are nice to know but not necessarily directly linked to programmatic decisions. When designing an entomological surveillance system, malaria programs should be clear about the decisions they have to make and the information needed to make the best decisions. When countries have achieved very low levels of transmission and are approaching elimination, it is likely that they are operating with the existing entomological surveillance system that contributed to their progress in controlling malaria. In addition to the existence of sentinel sites and the capacity to conduct laboratory procedures, the country is also likely to have a good historical understanding of the vectors incriminated in malaria transmission in the past, variation in vectorial capacity across the country, and the vector's typical behaviors. As a result, most countries do not need to build an entomological surveillance system from scratch. Rather, the challenge for most countries will be determining how to transition an existing system to one that can provide the type of information that is needed in elimination settings. For the purpose of this training, however, we will describe the approach designing an optimal entomological surveillance system for an elimination setting as if there has not been an existing system. We have already reviewed the first two steps in designing an entomological surveillance system, that is, identify the key programmatic decisions, determine which entomological indicators are required. Once the key indicators have been agreed upon, you will need to decide how and when to collect the entomological data, including what methods to use and the period of the year when you should collect the data. Depending on the indicator and the question, you may need to establish sentinel sites to monitor trends over time, or you could select areas of the country to be sampled at some frequency without establishing fixed sites. You will need to develop data capture and data management instruments. These could be digital or paper-based in the field, but ultimately they need to lead into digital systems for analysis. It is also important to develop an analysis and dissemination plan in advance to ensure that the data collected and the form in which they are digitally recorded can easily feed into the analysis and dissemination approach. Finally, technicians will need to be informed of the indicators to be collected and the reasons for their collection, and trained in the methods for collecting entomological data and capturing the information in data systems. After the training, the system can be implemented, monitored, evaluated, and data analyzed. As a general rule of thumb, the quality of the data collected should take precedence over the amount of data collected. First, you should ensure that a few sites are collecting high-quality data that are useful for making programmatic decisions before expanding to the collection of additional indicators or to new sites. The main types of data collection that should be undertaken in areas approaching elimination involve longitudinal data collection, spot checks, investigations, and resistance assays. Most of the indicators we reviewed would be collected longitudinally at fixed sites, i.e. sentinel sites, located in areas that represent the range of eco-epidemiological settings in a country, including ecological zones with different malaria vector species and different levels of transmission. By continuing routine collection of entomological data at these sites over time, temporal changes can be monitored. By ensuring a representative distribution of sites, some of the heterogeneity and vector characteristics can be captured. Longitudinal data collection at fixed sites helps to identify and implement needed changes in vector control strategies. This should be conducted in areas where vector control interventions are implemented, areas with the risk of introduction of new species, and areas with rapidly changing environments. As transmission declines, the location of sentinel sites should be reevaluated to ensure that areas of ongoing transmission or high malariagenic potential are monitored. The timing and frequency of data collection at fixed sites will depend on several factors, including the specific indicator and how important variability throughout the year is for the decisions to be taken with those data. However, most longitudinal data collection sites will monitor species-specific vector density several times each month. The availability of mosquito larval habitats might also be monitored on a monthly basis, along with measurement of the presence or absence of larvae at these sites. More extensive surveys of vectoral behaviors might be focused during on months with peak mosquito densities in order to optimize the number of mosquitoes that can be observed. Spot checks are ad hoc cross-sectional assessments carried out in selected locations at specific points in time. Their purpose is to provide answers to specific and sometimes urgent programmatic questions, such as confirming suspected problems in the quality of implementation of an intervention, expected increases in receptivity, or changes in the environment that might alter the vector distribution. In the absence of routine entomological surveys, they help to evaluate changes in the risk of malaria reintroduction derived from resettlement, migration, changes in economic activity, or in environmental changes, or to confirm the suspected introduction of new vector species. Outbreak or focus investigations are similar to spot checks in that they are implemented only when needed. However, outbreak or focus investigations are undertaken when there is new, resurgent, or persistent transmission, and it is unclear what factors are driving transmission. As we will review in Unit 5E on focus investigations, entomological investigations are only conducted if needed to confirm or refute one of the hypotheses developed in advance. The timing and frequency of spot checks and investigations is entirely determined by need. Finally, resistance assays may be conducted fixed laboratory sites, but may use mosquitoes collected from sentinel sites, spot checks, or outbreak or focus investigations. Resistance assays determine the susceptibility of primary and secondary vectors to insecticides used in vector control. It is beyond the scope of this course to go into detail on the methods used to collect entomological data, but we will briefly review the main techniques so that you are familiar with the types of methods commonly used. Some of these methods can contribute to more than one indicator. Adult mosquitoes are often collected using human landing catches, wherein one or more collectors bare their legs and arms and use aspirations to collect any mosquitoes that land on them. This method can be used to determine the presence or absence of malaria vectors in the area, derive human biting rates, determine peak biting times, and compare biting rates indoors and outdoors. Mosquitoes that are collected can be analyzed in a variety of ways to derive other indicators. Collectors need to take malaria prophylaxis to ensure they are not infected as a result of their activity, and the method might not be ethical in areas with evidence of arbovirus disease transmission, in conflict areas, or in areas of significant civil unrest. CDC light traps attract adult mosquitoes seeking blood meals at night. They are typically hung next to sleeping residents who are protected by mosquito nets. The number of mosquitoes caught in light traps can be converted to derive the indoor biting rate, and traps can be hung outside to measure the outdoor biting rate. To capture resting mosquitoes, aspirators can be used to collect mosquitoes resting on indoor surfaces. However, it can be challenging to collect resting mosquitoes when mosquito densities are very low. Finally, to determine whether water bodies are mosquito larval habitats and contain larvae or pupae, dipping can be used. This is a simple and inexpensive method that may be more effective in small larval habitats and biased towards the edge of larger water bodies. The slide summarizes the main surveillance methods for collecting mosquitoes, but there are many others. At the end of the session, references are provided for those interested in learning more. We have discussed the importance of conducting insecticide susceptibility tests in areas where insecticide-based vector control is implemented. Decisions on the insecticide to be used in IRS and whether or not ITNs with PBO should be deployed are based on monitoring malaria vector susceptibility to insecticides. There are two standard and complementary procedures for measuring susceptibility in adult mosquitoes: the WHO tube test and the WHO bottle bioassay. Until now, the only available WHO standard susceptibility test procedure was the WHO tube test. This test involved exposing adult mosquitoes to papers impregnated with a discriminating concentration of insecticide in specially designed plastic tubes. However, the chemical properties or modes of action of some insecticides prevent their impregnation on filter papers. To enable monitoring of vector resistance to these insecticides, a new WHO standard procedure was developed and named the WHO bottle bioassay. This should not be confused with the CDC bottle assay, which uses different durations of exposure and measures mosquito knockdown rates at the end of the exposure time. The basic procedure, whether using the WHO tube or bottle assay, goes like this. Immature mosquitoes are collected from areas where insecticide-based vector control is implemented. Larvae and pupae are reared to adulthood. Adult mosquitoes are exposed to known concentrations of an insecticide for a fixed period of time. The number of dead mosquitoes is recorded at least 24 hours after exposure. The proportion of mosquitoes killed is used to determine whether or not resistance is present. This assay does not provide information on the mechanism of resistance, only whether or not it is present. The classification of mortality data from resistance assays is shown on this slide. If 98% or more of the exposed mosquitoes die, then they are classified as susceptible to that insecticide. If less than 90% are killed, resistance is confirmed. In the intermediate range between 90% and 97% mortality, the test should be repeated with untested mosquitoes from the same population or progeny of mosquitoes that survived the bioassays. The data from resistance assays should be reviewed by the program on a regular basis to decide whether action should be taken with respect to choice of vector control interventions or change in insecticides. We will take a few minutes to have small group discussions regarding entomological surveillance in your area or country. Please form small groups and discuss these three questions. Does your country or area have an entomological surveillance plan? Does the plan identify the key programmatic questions? If not, what do you consider the key questions to be? Does the plan clarify which indicators should be measured and how they contribute to decision making? If not, which indicators do you consider to be the most important? How would you use the indicator to make programmatic decisions? A core component of a malaria program is monitoring and evaluating the impact of interventions, including vector control, to ensure they are implemented with good quality. Let's review the components of quality assurance and monitoring and evaluation of vector control. As for any programmatic activity, there should be a quality assurance plan for vector control. The first step in ensuring a quality intervention is with the procurement of ITNs, insecticides, or larvicide. WHO has a pre-qualification department that evaluates and lists efficacious products that meet good manufacturing practice standards. National malaria programs should purchase products pre-qualified by WHO or that have gone through their own national regulatory processes. The next step in a quality-assured vector control program is to ensure that the intervention is implemented appropriately. Good micro-planning is needed to ensure that the appropriate quantities of ITNs, insecticide, and larvicide are available in the right places for deployment. High-quality training is needed to ensure that staff distributing ITNs, spray operators, and vector control staff have the necessary knowledge and skills to perform the tasks to which they are assigned. It is critical to have an effective supervision system in place. Supervision is particularly needed to ensure the quality application of insecticides for IRS and larviciding. Routine program monitoring is needed during campaigns to monitor coverage of each strategy to ensure that program goals are met. In the following slides, we'll review how to monitor the performance of recommended vector control interventions to ensure they are working as intended. ITNs are highly effective means of preventing malaria infection and reducing malaria transmission. In populations provided with ITNs, they have been shown to reduce all-cause child mortality by 20%, decrease clinical malaria cases by 50%, and severe malaria cases by 45%. The ITN use by children in the studies that produced these estimates was typically above 60%. ITNs have also been shown to reduce malaria in pregnancy and improve infant birth weight. ITNs should be purchased from the WHO list of pre-qualified ITNs. WHO undertakes a rigorous evaluation of the safety and effectiveness of each product and supports quality control testing. The important aspects to monitor and evaluate with ITNs related to their performance are indicators of correct and consistent use, vector susceptibility to pyrethroids, and net durability. Indicators of correct and consistent use are generally collected in household surveys and include the proportion of households with at least one ITN for every 2 people and the proportion of individuals who slept under an ITN the previous night. Reports of low access to ITNs can be addressed through better planning and implementation of large-scale ITN distributions, while low rates of use might be addressed through social and behavioral change communication. We have already spoken about the need to monitor vector susceptibility to pyrethroids to determine whether an area should switch to pyrethroid PBO nets. Finally, manufacturers generally claim that ITNs last for 3 years, but there is significant variability in net durability depending on the type of ITN, the type of structures in which they are deployed, and wear and care. Durability monitoring may not be as necessary in an area approaching elimination that is not deploying many ITNs, as it may be in higher transmission areas with many ITNs. However, if there is a concern that ITNs are not lasting as long as they should, programs should develop a durability monitoring program and implement it within the next mass distribution campaign. The durability monitoring program should include aspects of attrition, physical integrity, insecticidal activity, insecticide content. Durability data can inform replacement strategies and behavioral change activities aimed at increasing ITN longevity and impact. IRS is the organized spraying of the interior surfaces of houses. It can be conducted prior to the peak malaria transmission as proactive IRS or in reaction to a confirmed case as reactive IRS. IRS kills adult female mosquitoes when they rest on the sprayed surface after feeding. IRS might also have a repellent effect and deter mosquitoes from entering the house. IRS has been used for many decades as an effective vector control tool that has an overall impact on reducing mosquito densities. The quality of IRS spraying is monitored in standard WHO cone assays conducted immediately after spraying and once a month thereafter during the expected duration of residual efficacy of the insecticide formulation. Similar to the way resistance is monitored, adult mosquitoes are introduced into the cone and exposed to a sprayed surface for a fixed amount of time. The number of mosquitoes killed or disabled by the insecticide is counted and the proportion killed is calculated. Any concern about poor quality should be relayed to the operational teams immediately. Remedial measures will depend on the findings of investigations of spraying quality. They may include more closely supervising the spray teams, retraining the spray operators, verifying the quality of the IRS products used, or respraying houses in the target area. When larviciding is used, its impact should be determined by monitoring changes in vector density before and after implementation. Currently, there is no guidance on how to monitor the effectiveness of house screening. We will break into small groups to discuss some questions around monitoring and evaluating vector control in your area or country. After 15 minutes, we will reconvene in plenary to discuss these questions. If ITNs are deployed in your area, what are some of the problems you have encountered with their performance? What data could help you understand why those problems are occurring? If ITNs are deployed in your area, is durability monitoring implemented? Are there any issues with ITN durability? If IRS is used in your area, does the malaria program conduct cone assays to monitor the bioefficacy of the spray? If IRS is used in your area, are insecticidal choices made based on a review of the data from resistance bioassays? In summary, selection of entomological indicators and design of the surveillance system should be based on important programmatic questions. 2. In elimination and post-elimination settings, the critical indicators are vector presence and absence, species composition, and resistance frequency and status. If larviciding is used, then habitat availability and occupancy are also important. 3. Entomological surveillance systems may consist of sentinel sites, spot checks, outbreak or focus investigations, and resistance assays. 4. The effectiveness of ITNs, IRS, and larviciding should be monitored and evaluated, and corrective measures taken if performance is not as expected.